Hello, my dear students. Have you ever wondered how every cell in our body gets oxygen and nutrients, even the one in your toes? The circulatory system is like a delivery service. The heart is the engine, blood is the vehicle, and vessels are the roads. It carries oxygen, nutrients, hormones, and removes waste from our body. Today, in this class, we will learn how the heart works, how blood flows, and what each blood vessel does, and why maintaining a healthy heart is important to us. Most organisms, including humans, have circulatory body fluids that act as both distributing and collecting system. These body fluids help deliver substances like nutrients and oxygen and collect waste products from all other parts of the body, reaching even the most distant cells. So in this class we will focus on the composition and functions of these circulatory fluids need for transport inside the body. So we can see when a blood moves through a blood vessel, it absorbs nutrient from food which are transported to all body cells for energy, growth and repair. Oxygen is taken in through the lungs and delivered to cells, while carbon dioxide waste is carried back for exhalation. Excretory system, it removes water, salts and urea by transporting them from cells to excretory organs for elimination from our body. It also produces hormones. Endocrine system produces hormones that are carried by blood to regulate body functions. Circulating fluids, blood and lymph transport materials and perform other vital functions. This slide illustrates the double circulation in the human body, showing how deoxygenated blood, blue color, from the body is pumped to the lungs for oxygenation, and how oxygenated blood, which is red in color, returns to the heart and is then circulated to the entire body. It highlights the systemic and pulmonary circulation and the distinct pathways for oxygen rich and oxygen poor blood. Fluids in our body, principal circulatory fluids. Our body has three main circulating fluids. Blood, it is found in the heart and blood vessels. It is part of our circulatory system. Tissue fluid, it fills the spaces between the cells and in organs. It is derived from the blood plasma. Lymph, it is present in lymph vessels and lymphatic organs like the spleen and tonsils. Blood circulation, it circulates in a closed manner within vessel. It is a closed circulatory system. It carries oxygen, nutrients, hormone and waste products. Pulmonary artery basically carries deoxygenated blood from heart to lungs, and pulmonary vein returns oxygenated blood to the heart. Lymph and tissue fluid relationship. Blood plasma leaks out of blood capillaries forming tissue fluid. Excess tissue fluid enters lymph capillaries becoming lymph. Lymph flows through lymph nodes and re-enters the blood via the lymphatic duct. This is the color differentiation. Arteries are shown in red, which carry oxygenated blood, and veins are shown in blue, which carry deoxygenated blood. Through actual deoxygenated blood is dark red. Veins appear blue due to light reflection and thinner walls of the blood vessel. Open and closed circulatory system. Closed circulatory system, blood stays within vessels just like humans, and in open system in animals like insects, blood flows freely in body cavities without vessels. Non-circulating fluids, certain body fluids which do not circulate, just like synovial fluid which is present in our joint cavities, and vitreous humor which are present in the eye. Now we'll study properties of the blood. It is never stationary. Blood constantly circulates from the heart to arteries and returns via veins. Its color is blood is thick. Blood is basically thick, bright red color when taken from arteries. It is oxygenated blood, and when it is dark red when it is taken from veins, it is actually deoxygenated blood. An average adult human has 5 to 6 liters of blood by volume. Blood test salts even experienced during cuts or gums bleeding. pH level, blood is basically or slightly alkaline with a pH between 7.3 and 7.45. Here the neutral pH is 7. Functions of the blood: transport by blood digested food, oxygen, carbon dioxide, excretory materials. We have already studied that. Hormones and heat also is distributed to maintain uniform body temperature. So this is the function. Transport is one of the main function of the blood. Then protection by blood, it is responsible for clot formation. It prevents blood loss and stops entry of germs and pathogens. WBCs protect by engulfing pathogenic bacteria. Blood also produce antitoxin and antibodies which neutralize the poisonous substances or kill the germs. Composition of blood. Blood is a fluid connective tissue composed of two main components, plasma and cellular element. Plasma is the liquid part of blood, making up about 55% of total blood volume. Its composition: it has water, it acts as a solvent and medium for transport. Proteins: it includes albumin, globulin and fibrinogen. Solutes such as nutrients, glucose, amino acids, lipids, vitamins. Waste product হচ্ছে urea, uric acid, creatinine. Gases: oxygen, carbon dioxide, nitrogen in small amounts. Hormone and other enzymes. Electrolytes like sodium, potassium, calcium, chloride, bicarbonate. Cellular elements: these make up of about 45% of blood volume and include red blood cells, white blood cells and platelets. Red blood cells are also called as RBCs or erythrocytes. White blood cells are called as WBCs or leukocytes, and platelets are called as thrombocytes. RBCs are most abundant cells. They are basically biconcave, anucleated in case of adult mammals, and it is flexible in nature. It contains hemoglobin, which is a red pigment and binds oxygen and carbon dioxide. Main function of RBC is transport of gases like O2 and CO2. Its lifespan is about 120 days. But WBCs is larger than RBC but fewer in number than RBCs. It has nuclei and are involved in immune defense. There are basically two types of WBCs: granulocytes and agranulocytes. Granulocytes are the acidophilic granules like neutrophils, eosinophils and basophils, but agranulocytes have no granules. There are also two types: lymphocytes and monocytes. Platelets are small disc-shaped non-nucleated fragments. They help in blood clotting by releasing clotting factors. Their lifespan is about 8 to 10 days. This slide shows the formed elements of blood, including red blood cells, white blood cells like monocyte, lymphocyte, eosinophil, basophil, neutrophil, and the platelets, each with their distinct structure and function. Life and death of RBC. Now we will study what is how RBC is formed. The process of formation of RBC is called as erythropoiesis. It is produced in red bone marrow, especially in long bones like ribs, sternum and pelvis. The process is called as erythropoiesis. It is stimulated by a hormone, erythropoietin, which is secreted from the kidney in response to low oxygen level in blood. RBC develop from pluripotent hematopoietic stem cell through several stages like proerythroblast to reticulocyte to mature RBC. So reticulocytes are released into the bloodstream and they mature into RBCs within 1 to 2 days. Structure and characteristics of RBC. Mature RBCs are basically biconcave anucleate cells allowing maximum surface area for gaseous exchange. Their diameter is about more or less 7.5 micron and thickness is nearly 2 micron. It contains hemoglobin which binds oxygen and carbon dioxide. They lack nucleus, mitochondria, endoplasmic reticulum to accommodate more hemoglobin. The lifespan of an RBC is about 120 days in human circulation. During this time, RBCs repeatedly pass through narrow capillaries, undergo mechanical stress. Senescence or aging. As RBCs age, they become less flexible and their membrane proteins deteriorate. Aged RBCs are recognized and removed by the reticular endothelial system, especially in the spleen, liver and bone marrow. Destruction or hemolysis. Old or damaged RBCs undergo phagocytosis by macrophage in the spleen, called as graveyard of RBC. Their hemoglobin is broken down. Globin part is the amino acid which is used for protein synthesis. Heme part, it is broken into iron and Fe2 plus iron. It is recycled and stored as ferritin or transported via transferrin. Then the second part of the heme is the porphyrin ring. It is converted to bilirubin, which is transported to the liver and excreted via bile. Clinical relevance. Excessive destruction of RBCs causes hemolytic anemia. Defective production causes aplastic anemia, thalassemia, sickle cell anemia, and bilirubin accumulation can cause jaundice. Hemoglobin, it is a conjugated protein made up of hemine and globin found exclusively in RBCs. It is essential for respiratory gas transport in vertebrates. The structure composed of 4 polypeptide chains. Globin part contains 2 alpha and 2 beta chains in adults, hemoglobin A, and each chain is bound to a heme group which contains a central iron atom, that is Fe2 plus. Each iron can bind 1 molecule of oxygen, so one hemoglobin molecule can carry up to 4 oxygen molecules. Types HbA হচ্ছে adult hemoglobin, major types in adults more or less 97%. HbF হচ্ছে fetal hemoglobin, higher oxygen affinity and replaced by HbA after birth. HbA2 it is the minor adult form. Abnormal types হচ্ছে HbS in sickle cell anemia or HbC etc. Oxygen transport in lungs, hemoglobin combines with oxygen to form oxyhemoglobin. Hb plus O2 it will form the HbO2. In tissues where oxygen pressure is very low, hemoglobin releases oxygen for the purpose of cellular respiration. Now we'll go for carbon dioxide transport. In tissues, hemoglobin, the protein can bind to carbon dioxide to form carbaminohemoglobin, which is HbCO2. Hb plus CO2 it will form HbCO2. Only about 20 to 25% of carbon dioxide is carried by this process, and the rest is transported as bicarbonate ions, which or it can be transported through dissolved in plasma. Now I'll go for the color of blood. We have already mentioned that bright red it is when oxygenated, that is when oxyhemoglobin is formed, and dark red when deoxygenated, that is reduced hemoglobin. This difference in color is the basis for showing arteries as red and veins as blue in diagram, though deoxygenated blood is not truly blue. Physical importance facilitates oxygen delivery to all body cells. It plays a role in buffering blood pH. It removes carbon dioxide, a metabolic waste. Normal hemoglobin main 13.5 to 17.5 gram per deciliter, women হচ্ছে 12 to 15.5 gram per deciliter, children থাকে 11 to 16 gram per deciliter. Levels may vary based on age, sex, altitude and health. Clinical evidence: low hemoglobin indicates anemia causing fatigue, pallor and breathlessness. High hemoglobin may occur in polycythemia, dehydration or high altitudes. It is measured using complete blood count or CBC test in diagnostic centers. Origin and life of RBCs or origin and life of WBCs or leukocytes. WBCs are formed in red bone marrow like the RBC and from hematopoietic stem cells. Obviously, the process of WBC is called as leukopoiesis. There are two main types of WBC: granulocytes and agranulocytes. Granulocytes are neutrophil, eosinophil and basophil, and agranulocytes are lymphocyte and monocyte. They are less visible. Site of maturation: granulocytes and monocytes mature in bone marrow, but lymphocytes and mature in B lymphocytes, that is bone marrow, and T lymphocytes in thymus. Circulation and migration: WBCs can circulate in the blood for a short time. They migrate into the tissue through diapedesis to perform immune functions. There are different types of WBCs, or granulocytes or agranulocytes. The lifespan: neutrophils lifespan is 6 hours to a few days. Lymphocytes several days to years, especially memory cells. Monocytes হচ্ছে a few days in blood, longer in tissue as macrophages. Eosinophil or basophil হচ্ছে few days. So when we found the pluripotent stem cell, that is basically myeloid stem cell and lymphoid stem cell. Myeloid stem cell will produce various precursor or blast cells like which will produce neutrophil, eosinophil, basophil, monocyte, platelet and erythrocyte, and only lymphoid stem cell will produce the blast cells to join the lymphoid cell. So and function: defend the body against infection, allergen and toxic and tumors. It performs phagocytosis and produce antibodies and mediate immune responses. Fate: WBCs complete their role and die via apoptosis. It is programmed cell death or removed by macrophages, constantly replenished to maintain immune defense. Diapedesis of WBC: diapedesis is basically the process by which white blood cells or WBC squeeze through the walls of blood capillaries into the surrounding tissue. Purpose: it enables WBCs to reach the site of injury, infection or inflammation, allowing WBCs to actively defend the body by engulfing pathogens, which is called as phagocytosis, or relieving immune responses. Importance: it is crucial for immune defense, fights pathogen for more efficiently at the infection site. It promotes healing by removing dead cells and debris. Leukemia: leukemia is a type of cancer of the blood forming tissue, include the bone marrow and lymphatic system, characterized by abnormal and excessive production of immature or defective white blood cells. Leukopenia: leukopenia is a medical condition marked by an abnormally low white blood cell count in the blood, which reduces body's ability to fight infections. Cell type: there are different types of cells. I mentioned their nuclear morphology, the granules present in them, their differential count percentage, their lifespans and their functions also. Functions of leukocytes: We have already mentioned the main functions are phagocytosis, inflammation, formation of antibodies, formation of antitoxins, etc. Functions of platelets or thrombocytes, its mainly function is blood coagulation. The injured tissue cells and the platelets which disintegrate at the site of the wound release a substance called thrombokinase, or also called as thromboplastin. The thrombokinase acts as an enzyme with the help of calcium ion present in the plasma to it converts a substance prothrombin of the plasma into the thrombin. Vitamin K and fat soluble vitamin is essential for the production of prothrombin. Thrombin in the presence of calcium ion reacts with the soluble fibrinogen of the plasma to convert it into an insoluble fibrin. Fibrin is a solid substance that forms threads. These microscopic threads of fibrin are sticky and form a network or mesh-like structure at the wound. Blood cells are trapped in the network of the fibrin. The network then shrinks and squeezes out of the rest of the plasma, which is in the form of a clear liquid, the serum. The solid mass which is left behind is called clot or thrombus. Now we'll study hemophilia. It's a genetic disorder in which the blood lacks sufficient clotting factors, leading to prolonged bleeding. It is usually inherited and caused by a deficiency of factor 8, which is known as hemophilia A, and if it causes due to absence of factor 9, it is called as hemophilia B. Even minor injuries can result in excessive bleeding, especially into joints and muscles. Hemophilia is typically an X-linked recessive disorder, primarily affects the male. Now let's go for the differences between erythrocytes, leukocytes and thrombocytes. Now please go through it. Now let's study blood transfusion. The process of transferring blood or blood components from one person, that is donor or recipient, it is called as blood transfusion. It is used to treat blood loss due to injury or surgery, anemia, clotting disorders or certain diseases. Compatibility is crucial. Incompatible transfusion can cause agglutination or jumping, and hemolysis, destruction of RBCs, which can be fatal. Blood cross matching, performed transfusion to induce compatibility between donor and recipient blood. ABO blood group system, it was discovered by Karl Landsteiner in 1901. The basis was presence or absence of antigen A and antigen B on the surface of red blood cells. Group A blood, it has A antigen on the RBCs and anti-B antibodies in the plasma. Group B has A antigen in the RBCs and group B has the plasma. Then group AB, it has A and B antigen on RBC, but though there no antibodies are present in plasma. Then lastly group O, which has no antigen on RBC and both anti-A antibodies in plasma are present. So we will found the universal donor are O negative and universal recipient is A positive. Importance of transfusion, incompatible transfusion can cause agglutination or clumping and hemolysis of RBCs, which is very much life threatening. Rh blood group system. Rh is a type of protein on the outside of the red blood cell. Landsteiner and Weiner in 1940 discovered it. It is also known as D antigen. RBCs have the D antigen called as Rh positive, and RBC lack the D antigen called as Rh negative. Rh incompatibility occurs when Rh negative mother carries Rh positive fetus, leading to the hemolytic disease of the newborn. Erythroblastoma fetalis can happen in subsequent pregnancies. Prevention. Injection of anti-R antibodies or Rho-D or immunoglobulin to the Rh mother after the birth of Rh positive. As a compatibility in transfusion, Rh negative individuals should receive only Rh minus blood. Rh positive individuals can receive both Rh minus and Rh positive blood, but Rh positive is preferred here. Blood circulatory system. The circulatory system consists of heart, blood and blood vessels like artery, vein and capillaries. The heart is a central muscular hollow concept organ about the size of a closed fist, located slightly left of the middle in the thoracic cavity within the mediastinum. It is enclosed in a double layered sac called pericardium, which provides protection and reduces friction during contraction. The heart is basically four chambered, two upper atria, thin walled chambers that receive blood, and two lower ventricles, thick walled chambers which can reduce the blood. The right side of the heart handles deoxygenated blood and the left side handles oxygenated blood. The heart wall has three chambers: epicardium, myocardium and endocardium. Epicardium is the outer layer, myocardium is the middle muscular layer, and endocardium is the inner lining of the chambers. The heart functions through rhythmic contraction and relaxation. Systole is the contraction phase and diastole is the relaxation phase. This slide illustrates the structure and internal anatomy of the human heart, showing labeled parts such as chambers, valves, major blood vessels and heart valves, along with the direction of blood flow through heart and lungs. Blood vessels entering and leaving the heart. Vessels entering the heart are veins. There are mainly three types: superior vena cava, inferior vena cava, and pulmonary veins. Superior vena cava and inferior vena cava, they brings deoxygenated blood from the upper and lower side of the body respectively, and pulmonary veins to carry two from each lung. দুটো লাং থেকে একটা করে ক্যারি করবেন। Vessels leaving the heart or arteries. The pulmonary arteries from the right ventricle carries deoxygenated blood to the lungs for oxygenation. The only artery that carries deoxygenated blood. Aorta originates from the left ventricle, the largest artery in the body. Distributes oxygenated blood to all parts of the body through its branches. Now coronary arteries. The coronary arteries are the blood vessels that supply oxygen-rich blood to the heart muscle or myocardium itself. They arise from the base of the aorta and include left coronary artery and right coronary artery. Their proper function is vital for the heart's health and performance. And now we'll go for myocardial infarction. It is also called as heart attack. The myocardial infarction is the death of heart muscle tissue and to a prolonged lack of oxygen. But in case of angina pectoris, it is a condition marked by temporary chest pain or discomfort due to reduced blood flow to the heart muscle. It is usually triggered by physical exertion or emotional stress and relieved by rest or nitroglycerin. Now, unlike myocardial infarction, angina does not cause permanent damage to the heart. It is considered as a warning sign of underlying coronary artery disease. Valves regulating unidirectional blood flow, anteroventricular or AV valve. There are two types of AV valve: right tricuspid, which is located between right atrium and right ventricle. It has three cups, so it is called as tricuspid. It prevents backflow of blood into the right atrium during right ventricular contraction. Bicuspid or mitral valve. It is located between the left atrium and left atrium or left ventricle. It has two cusps present. Backflow of the lung prevents backflow into the left atrium during left ventricular contraction. Chordae, tendineae and papillary muscles, these structures anchor the AV nodes and prevent them from inverting during the ventricular contraction. Semilunar valves, pulmonary semilunar valve, it is located at the base of the pulmonary artery between the right ventricle and pulmonary artery, prevents backflow of blood into the right ventricle after contraction. Aortic semilunar valve, which is located at the base of the aorta between the left ventricle and aorta. It prevents backflow into the left ventricle after ejection of the blood. Function of valves: all valves ensure NC2, that is unidirectional flow of blood through the heart, and prevent হ্যাঁ তাহলে কি বলছে function of valves: all valves ensure one way or unidirectional flow of blood through the heart and prevent regurgitation or backflow. These opening and closing are passive and governed by pressure differences across the chambers. Circulation of blood in the heart: two main phases of heartbeat: atrial systole. The SA node initiates the electrical impulse causing the atria to contract, and deoxygenated blood from the body enters the right atrium via superior vena cava, inferior vena cava and coronary sinus. Oxygenated blood from the lungs enter the pulmonary artery. তাহলে কি বলছে artificial contraction passes into the আচ্ছা tricuspid valve and bicuspid valve. AV valves are opened during this phase and ensuring smooth flow from atrial to ventricles. So this is the diagram. This is the showing the flow of blood within the heart. Ventricular systole: the impulse reaches the AV node, then travels via the middle length of the heart, and it is via the bundle of His and Purkinje fiber triggering ventricular contraction. AV valves close to prevent backflow into the atria. It is called as LAB, and the right ventricle pumps deoxygenated blood to the pulmonary artery via the pulmonary valve towards the lungs. The left ventricle pumps deoxygenated blood into the aorta via the aortic valve supplying the entire body, and lastly verticals empty. The semilunar valves close, preventing the backflow of water. Now We'll go for the heartbeat. A heartbeat is one complete contraction and relaxation of the heart muscles, both atria and ventricles. It is responsible for pumping blood through the lungs and body in rhythmic manner. The average human heart beats 72 times per minute at rest. A heartbeat involves two main phases, systole and diastole. Systole is the contraction phase, first atria then ventricles. Diastole is the relaxation phase. The electrical impulse originates from the AC node or sinoatrial node, spreads through the AV node, atrioventricular node, and travels through the bundles of His and Purkinje fiber. This ensures coordinated contraction. Atria contract first and followed by ventricles. Heart sounds. The heart produces two main types in each cardiac cycle, such as lab and dub. First heart sound, lab, it is caused by the cause of atrioventricular valves. Tricuspid and bicuspid or mitral valves occurs at the beginning of the ventricular systole. It is a longer and louder sound. Second heart sound, dub, it is caused by closure of semilunar valves, pulmonary and aortic valves. Occurs at the end of the ventricular systole, beginning of diastole. It is shorter and softer than LUB. Pacemaker. The pacemaker is a group of specialized cardiac cells that regulate the heartbeat by generating rhythmic electrical impulses. The AC node located in the right atrium acts as natural pacemaker, including initiating impulses at 70 to 75 beats per minute. These impulses travel through the AV node, bundle of His, Purkinje fibers, coordinating heart contractions. An artificial pacemaker is a medical device that implanted under the skin to correct abnormal rhythms like bradycardia, heart block, etc. It ensures normal heart rate and rhythm. It prevents impaired contraction and relaxation and cardiac arrest. Blood vessels, basically arteries, it carry oxygenated blood away from the heart, except pulmonary artery, which only carry deoxygenated blood. Artery have thick elastic and muscular walls to witness high pressure. It has lumen, inner space is narrow. It blood flows under the high pressure in spurts. It do not have valves at the pressure, prevents backflow. Located deep inside the body, veins, it carry deoxygenated blood towards the heart, except pulmonary vein, which carry oxygenated blood. It has thin, less elastic wall with less smooth muscles. Lumen is wider in arteries. Blood flows under low pressure and more steadily. It contains valves to prevent backflow of blood, often located closer to the skin, which is superficial. Functions of capillaries: exchange of gases, nutrient delivery, waste removal, hormone transport, thermoregulation, connecting link, selectively permeable, blood pressure regulator. These are the functions of blood capillaries. Circulation of blood or double circulation: deoxygenated blood flow, pulmonary circulation. Deoxygenated blood from the body returns to the right atrium through superior vena cava, inferior vena cava, and coronary sinus. When the right atrium contracts, blood flows through the tricuspid valve into the right ventricle. On contraction of the right ventricle, this pulmonary valve opens and blood is pumped into the pulmonary artery. The pulmonary artery carries blood to the lungs, where carbon dioxide is released and oxygen is absorbed. अच्छा, so the human heart basically exhibits double circulation, pulmonary circulation, heart, lungs, heart. Systemic circulation, heart, body, heart. It involves the heart, ensure unidirectional flow and prevent backflow. The right side of the heart handles deoxygenated blood, and while the left side handles oxygenated blood. Circulation is coordinated by the cardiac conduction system, including AC node, AV node, bundle of His, Purkinje fibers. Hepatic portal system. The hepatic portal system is a special type of circulatory system that carries blood from the digestive organs to the liver before it enters the general circulation. The utility and function of the hepatic portal system is same as the RBC, that likes nutrient processing, detoxification, storage, metabolic regulation, filtration of pathogen, etc. The pulse. The pulse is the regular beating felt in the arteries when the heart blood pumps, especially during the heart's contraction, are called blood pressure. Pressure is the force with which heart pumps blood through the blood vessels. It has two readings: systolic pressure when the heart beats and pushes blood out. and the diastolic pressure when the heart rests between two beats. A normal blood pressure is about 120 by 180 by 80 mmHg. Tissue fluid. It is formed by the filtration of blood plasma through capillary walls. It bathes all body cells, supplying nutrients and oxygen, helps in exchange of substances between blood and cells. It lacks protein as they are too large to pass through capillary walls. Lymph, a clear colorless fluid derived from the tissue fluid, enters lymphatic capillaries and becomes lymph. Composition of lymph, it has water, white blood cells, proteins, fats and waste. Function of lymph, it returns excess tissue fluid to the body. It transport delivery wastes from the intestine to the blood and via the lymph vessels. Lacteals help in defense. It contains lymphocytes that fight infection. It removes cellular waste and drains interstitial spaces. Now I'll study the lymphatic system. It is a network of lymph vessels, lymph nodes and lymphoid organs like tonsil, spleen, thymus, etc. It collects lymph from tissues and returns to the blood via subclavian vein. Lymph nodes, it filter lymph and trap pathogens. It plays a crucial role in immune response and fluid balance. The spleen is a soft, dark reddish brown and the largest lymphoid organ located at the upper left side of the abdominal cavity, just below the diaphragm, and it is surrounded by a capsule and contains two types of tissues like red pulp and white pulp. The filtration of the spleen are following: filtration of the seed, immune response, filtration of the phagocytosis, storage of blood, and hematopoiesis. It is also found in fetus. During fetal life, it helps in formation of blood cells. So this chapter has been completed. Thank you, my dear students. All the best.